Hello, everybody. Welcome. Welcome to class, everybody. Hope everybody's doing well. Okay, yes, thank you. When you have a minute, just type your first name or your nickname into chat for me. Appreciate that. Very good. Just about everybody's here. All right, very good. Well, welcome. Welcome to another week. We are rapidly moving forward in the course. We're actually to the point now in the course where we make a bit of a switch in lecture and in lab. We've kind of gone through what you might think of as sort of introductory information about what microbes are and what they look like, how they're built, things like that. And we start taking more of a look into how they function, what their physiology is. So we're getting a little bit into some more complex material now. In laboratory, we're going to be doing experiments together that are a little more complex, and will take a little more of our critical thinking abilities to interpret the results that we generate together. So everyone, it seems like everyone is doing well so far. I'm pleased to say, and I think you're all ready to move forward. This week, here, let me pull this up. If you look at your syllabus in the back where the schedule is, this week your lecture topics include microbial biochemistry and microbial growth. Now, this biochemistry lecture, I think, is very difficult. This word, biochemistry, is basically the chemistry that relates to living cells, the type of chemistry that we who study living things need to understand and need to know. I recognize that a lot of people don't particularly enjoy chemistry. Some people really enjoy chemistry. Some people are very good at chemistry, but there are others of us who find chemistry very challenging. So just give yourself a little extra time this week as you go through the biochemistry material, and keep a list for yourself of anything that doesn't make sense to you, and we will try to talk about it. Of course, you can also message me privately about it if you'd rather, and I'll try to help you understand it that way. And don't forget, we also have that discussion board where you can post questions about material, and your classmates can chime in and try to be of help to you. This growth lecture, the second lecture for this week, this is specifically about how specifically bacteria divide, the process that they use in order to divide themselves, to create new cells. So again, it goes into a little more of the physiology of microbes versus more of the anatomy, which is really what we've been focused on so far. Now, a couple of you contacted me about My error, I didn't post the homework for you for the fungi lab. I actually posted it, but I didn't click where it made it public, so it wasn't visible to you, and I apologize for that. I thought I had double checked it, but I must not have. So I have remedied that now. There is a homework assignment posted for fungi, but because it's late, I've decided.
Hi, Mom.
I'm sorry. Because it's late, I've decided to make that homework assignment optional. So if you have the time and you'd like to do it, it's an opportunity to earn some points, obviously. I added an extra bonus point in as a little bit of encouragement for you. So it's possible to earn 7 out of 5 on this homework assignment, which is great. Little extra points here and there all add up. But if you don't have time to do it, don't worry. It won't count towards your grade if you don't do it. Okay. So again, I apologize for that. That was my error. All right. So don't think there was anything else specifically that I needed to mention to you, just that we do have your first draft of the pathogen project due on Wednesday this week. I've already received at least 1 or 2 of them. Thank you. So just remember, you're going to want to get that into me by Wednesday at midnight. I've had a couple questions about citations and when can I use a URL versus more of a complete citation. Don't spend too much time worrying about that. I would rather you spend your time right now making sure that your questions are full and complete and well written. If I need to, I'll make some notes for you regarding your citations and you can fix that on your final draft. Okay. So yeah, we'll be sure to get those into me by Wednesday at midnight. Does anyone have any questions that I can answer right now? Any questions about any of the lecture material that you might have taken a look at already, or?
I did have a question.
Yeah.
About so when you were talking about water, and I believe it was the second biochemistry
Yep.
Video, and there were the hydrogen electrons with the delta positives and
Yep.
the oxygen.
Yep.
I'm sorry, the hydrogen atoms, and then the oxygen atom with the delta positive.
That's right.
And then you were saying that there was a slight charge. Is that because like the size of the atoms? Because I even noticed that hydrogens are small and oxygen's big. So do the electrons just spend more time in the positive zone?
It's a strange concept, isn't it? This
Yeah.
idea that a neutral molecule like water doesn't have a charge, it's neutral.
Yeah.
But within the water, you've got this oxygen atom, which has a high electronegativity. And then you have 2 hydrogens that have low electronegativity. And remember, this word electronegativity, what that should mean to us is the ability to attract electrons. So what happens in the covalent bonds, and remember there's 2, there's one between the oxygen and the first hydrogen, and then there's another between the oxygen and the second hydrogen, 2 covalent bonds. And remember, all a covalent bond is a pair of electrons that are being shared, right? So if there's a covalent bond between an oxygen and a hydrogen, because of the difference in electronegativity, the oxygen is going to hog the electrons. The oxygen is going to pull them in. And because the electrons are just that little bit closer, they give that oxygen just a little bit of more negativity. So it's a partial negativity.
Does that promote like it's well, I guess I'm thinking about the plasma membrane and how it's hydrophilic.
Yeah.
Does that contribute to it?
So anything that has partial charges in it, like water does, is a polar molecule.
It is polar. Okay, that was my question.
So that's kind of how we define, if we want to call something polar, what we're saying is it's got partial charges in it. It has unequal electron sharing in it. And the heads on the outside of the membrane are polar, and the heads on the inside of the membrane are polar. It's the tail region in the center that is not polar. So there's no partial charges in those molecules. And that's what allows the membrane to exist. The fact that there's a center in that membrane that is hydrophobic, that will not mix with water. Because remember, there's all kinds of water outside the cell and all kinds of water inside the cell. And we need to divide those two compartments if we want the cell to survive. So it's that hydrophobic tail section in a membrane that keeps the water outside and keeps the water inside. And it's the polar heads that allow molecules to come up to a membrane and interact with a membrane. Because a lot of what's out there in the living world, the world of living things, is polar. Yeah, it's a lot of chemistry. And
that helped a lot. That makes sense.
Oh, good. I'm glad. It's a lot of chemistry. And if you never really, for some of us, our chemistry classes could be years ago, or so I understand that. But these are the sort of concepts that really do matter when we talk about microbes, because we're going to be talking about some of the inner workings of the cell. And some of this chemistry is really important because I'll be using the terminology. Now I'll be using terms like polar and non-polar and hydrophilic and hydrophobic and things like that. Good question. Anybody else? Anybody else have a chance yet to jump into that lecture? All right. Well, remember, if you have any questions as you're viewing it, just jot them down. And you can either ask me privately or we can talk about it as a class. All right, I'm going to go ahead now and I'm going to jump into our lab exercise for the day. Today, we're going to be talking about what's called special media. I love that we call it special. When we use that term, special media, it means this is not everyday growth media. This is not the kind of media that we constantly use in the laboratory. These are media that tend to be more used for particular diagnostic tests to help us, again, identify microbes that come into the lab. So I'm going to go ahead and pull up our slides here. I can close this little box over here. All right. So the special media today. I think special media are very interesting because, again, the results that we get from special media, we have to really think about. There are sometimes, it's almost like we're doing a puzzle when we interpret what's growing or not growing on special media. So our objectives today are, before we get talking about particular special media, we're first just going to talk about 6 types of culture media. We've already talked about a couple of these, but we'll add on to what we can classify as categories of culture media. We're going to spend a little extra time talking about the difference between selective culture media and what we call differential culture media. And we'll explain what we mean by those terms. And then we're going to have our experiment. We're going to compare the growth of a couple of particular microbes on 2 kinds of special media. One of them is called Mannitol Salts, Agar, and we abbreviate that MSA. And one of them is called eosin methylene blue Agar, and that one is shorthanded as EMB. So we will grow with the magic of remote. We will grow some bacteria on these plates, and and generate some results for ourselves. So we've talked about culture media very briefly in our labs so far. We said that culture media can be solid, it can be semi-solid, and it can be liquid, or what we call broth. Remember, if we're making broth, we're basically taking water and we're adding ingredients to the water. And we said that we can add a very short list of ingredients as long as those ingredients contain the things that all microbes must have in order to survive. We talked about how they have to have a protein source and things like that. So we can add ingredients to water and create a broth, or we can add ingredients to agar and make a solid medium. Remember, agar by itself is not nutritional. It's just the material that creates that smooth, solid surface for us. But one of the nice things about agar is that we can add ingredients to it before it solidifies, and we can feed the microbes with those ingredients. Now, the other thing to know is that you can turn a broth into an agar. All you have to do is add some powdered agar into your broth and you will create a solid media. So we're constantly sort of, I hate to say experimenting, but we're constantly sort of playing around with media in the laboratory when we need different forms of it under different circumstances. Remember, we said that we can classify all of these media, the liquids and solids and semi-solids, we can classify them as being chemically defined media or as being complex media. And we said the difference between those two is whether or not we know exactly what's in the media. So if we are putting in known ingredients, we can call that a defined media or a chemically defined media. If we are putting in ingredients that we don't know exactly what's in them, now we've created a complex media. So if we're going to add things like blood or some kind of a meat product or eggs or something like that, we don't exactly know every single molecule that's inside of those complex ingredients. So we would call that a complex media. Now, in a microbiology lab, there are lots of different kinds of media that are used. And it's all about what kind of microbes you're growing or you're trying to grow, and whether or not you're trying to figure out what that microbe is, or whether you're just trying to grow it. And each one of those many types of media has its own unique recipe. And as I said last time, a lot of these media you can purchase. You can purchase them as dried material that comes in a jar, essentially a plastic jar, and you can mix up the media in your lab. Or if you'd rather, you can buy media that's already been made. And it all depends upon your budgetary needs. Some of the media that we use, again, are just designed to help us grow microbes. So some of our media will allow virtually any kind of microbe to grow on it. They're just full of the kinds of ingredients that microbes love. And so we can grow bacteria on them, or we can grow fungi on them, or we can grow algae on them. Some media are just very general, and they'll allow virtually anything to grow on them. Some media encourage one kind of microbe to grow specifically. So there are some media that really are just for bacteria. There are some that are just for fungi and so on. Remember, we said Sabarodes media is a kind of media we use to specifically grow fungi. So the media is designed specifically for fungi. Now, some allow only what are called non-fastidious microbes to grow, while others support the growth of fastidious organisms. Here's some new terminology for us. This term fastidious means an organism, a microbe again, that is difficult to grow because they have some kind of strange special requirements nutritionally or perhaps environmentally. Think, for example, about a microbe that might need a very particular temperature in order to grow, or has a particular need for a very special kind of mineral. Those are called fastidious microbes. They're hard to grow. So we have to design media. If we're trying to grow them in the lab, we have to design media specifically for them. Other kinds of media are going to let non-fastidious organisms grow, the ones that aren't that hard to grow, that are basically organisms that can survive on a varied diet, essentially. Now, here's where here's something that we're introducing today in special media, which is some media are designed to support the growth of one type of microbe while specifically inhibiting another type of microbe. Now, when I talk about this, I'm saying within one group of microbes. So there are media that will let one species of bacteria grow or one genus of bacteria to grow while specifically inhibiting all other types of bacteria. So it's got ingredients in it that, you would expect bacteria to grow on, but something has been added or removed so that only that one genus or only that one species can grow. These are, again, what we tend to call the special media. And finally, there are a lot of media in microbiology that change color when things grow in them. And it gives us a visual clue that we've got one particular type of microbe growing. What we use to make this color change happen is a type of a dye called an indicator dye. Now, you may have heard of indicator dyes in chemistry class. These are dyes that will change color depending on the pH. And under certain circumstances, the media will begin when it has nothing growing on it at 1 pH. And then once microbes begin to grow in it and secrete wastes and so on, the pH will change and the media will change color. And it can be very useful to us because we can look at the plate and we can get an idea of what's growing on it. So we've got lots of different types of culture media available to us, designed for all different kinds of purposes, designed for growing specific things or non-specific things, designed for different types of microbes, designed for certain diagnostic tests, and so on and so on. Now, I want to talk about those 6 kinds of culture media. We've already talked about a couple of these, but we'll go through them all today so you have it all in one lab presentation. There's 2 terms on this slide, what we call basal media and what we call enriched media. Now, this is essentially what we've already talked about as chemically defined and complex. So when you hear the word basal media, it means exactly the same thing as a chemical defined media. Both of those terms mean the same thing. So anything like, as we said before, like a nutrient broth or something called a peptone water. These are recipes that are very simple in their ingredient list. Very short list of ingredients go into them. It's the minimal things that bacteria need to grow, and we call them basal or chemically defined. Now you'll notice these are media that are going to grow non-fastidious bacteria. These have to be, hardy bacteria that don't have any special picky needs in their diet. Now, if you compare that to enriched media, this term means the exact same thing as complex. or you may also read it called chemically undefined media. These are the media that have one of those ingredients or more, one or more of those ingredients that we just don't know everything that's in it. Blood, serum, egg. Some of them have egg yolk in them. Some of them have meat extracts in them. There's all kinds of exotic enriched media out there. One of the most common ones is called Luria Bertani broth, but nobody calls it this. Everybody calls it LB broth. This is an enriched media, very, very commonly used in microbiology labs. Now, the whole point of adding something like blood or egg yolk and creating one of these complex or enriched media is because you're trying to grow an organism that is fastidious. It's just hard to grow in basal media. So we give it enriched media instead. Now, in addition to the LB, which is probably the most common complex or enriched media used in the lab, another couple that you'll see are blood agar and what's called chocolate agar. Unfortunately, there is no chocolate in chocolate agar. All chocolate agar is blood agar that has been cooked. It has been heated up so that the blood actually cooks and it turns a chocolate color, which is how it got that name. Too bad it doesn't have any real chocolate in it. Now, over here on the left-hand side, I put this picture in for us because when you're looking at basal media, whether it's a broth or an agar, it tends to have this very basic beige kind of color. Remember, they have a very short list of ingredients, and whether it's a broth or an agar, they tend to have this very basic, very boring color. Over on the right-hand side, up at the top, this is what blood agar looks like. Blood agar, like the name suggests, has blood added into it. It's usually sheep blood. At least if you buy it commercially, it's sheep blood. Down at the bottom, this is chocolate agar. You can see why people call it chocolate agar. What they did was they took a blood agar plate or plates and they heated them up. They cooked the blood until it turned this color. Now you might ask, why bother doing that? Why not just leave it as uncooked? The answer is that when you cook things, right, you change their structure, and the difference in uncooked and cooked blood makes a difference for some bacteria. Remember, fastidious organisms basically are going to be put on blood or chocolate agar. Organisms that are a little hard to grow sometimes. And some of them might prefer the uncooked and some of them might prefer the cooked, depending on the species. So we always have our basal media or our chemically defined. And we have, and those are for non-fastidious organisms. And then we have our complex or enriched media for our fastidious organisms. But then we also have a lot of different kinds of special media, and that's what we'll talk about next. I mentioned to you that some of the special media are what we call selective, and some of them are what we call differential, and some of them are actually both. They are both selective and differential. So let's talk first about the selective media. Selective media are special media that allow the growth of some kind of a desired bacterium while inhibiting the growth of unwanted bacteria. Selective media are usually agar. They are usually solid. All right, I have a note here that there are selective broths, but we give them another name. I don't want you to worry about this other name because it's too easy to confuse enrichment media with enriched media. So don't even learn this term. Just think of them as selective. So if you think about the word selective, I want you to think, oh yeah, this is the kind of special media that can select which bacteria are going to grow on it. It's going to select one versus another. Now, one of the most common selective media in the lab is one called Mannitol Salt Agar. And again, this one is abbreviated MSA. So it contains mannitol, that's a carbohydrate, and it contains salt in the form of sodium chloride. And the salt is really important because the salt is at an amount that we don't encounter in other types of media. It can be 7.5% sodium chloride. You can even get some mannitol salt agar that has 10% sodium chloride in it. That's very high. Now, what's going to happen when you try to grow bacteria on mannitol salt is most of the bacteria aren't going to survive because salt is toxic to most bacteria at that 7.5%. And it's toxic to even more bacteria at 10%. If you think about it, historically, we have used salt to preserve food, right? In the old days, people used to salt things like meat. Before we had refrigeration available, they would store perishable meats in salt because it would prohibit spoilage of the meat. So salt is toxic to bacteria, generally speaking. So if you put 7.5% sodium chloride into this agar, this MSA, it's going to select for Staphylococcus species, especially two particular kinds. One is called Staphylococcus aureus, and one is called Staphylococcus epidermidis. That's quite a mouthful.
Are those the ones that grow or don't grow?
Yes, those are the ones that will grow. So remember, when you think about these selective media, think about that word select. The media is selecting. It's selecting Staph aureus and selecting Staph epidermidis and inhibiting all the other ones. It's going to inhibit Streptococcus from growing. It's going to inhibit all kinds of other bacteria from growing. I'm just going to mention here, just for your notes, that there are a couple of other coccus-shaped organisms that can grow on 7.5% sodium chloride, but they don't grow very well. They're going to grow really poorly, but I'm going to mention it. One of them is called Enterococcus, and one is called Micrococcus. Because of this, because 7.5% mannitol salt can grow two species of Staphylococcus, that's why they also make the 10% version, if you will. If I grow an organism on the 7.5%, I can't tell just by the salt concentration. I can't tell whether I've got Staph aureus or Staph epidermidis. If I want to select even more tightly, I can grow it on 10% sodium chloride, and only Staph aureus will grow on that. Staph epidermidis can't grow on 10%. So if I suspect that I have Staph aureus, or if I have a sample in the laboratory and I want to find out if there's any Staph aureus in it, an easy way to do that is to streak out those cells, that sample, onto mannitol salt agar, especially the 10% variety. Because if something grows on that 10% MSA, I know it's Staph aureus. nothing else can grow there. So it's very helpful. Now I'm going to say more about mannitol salt in a few minutes, because mannitol salt is one of the agars that is both selective and differential, but we'll get there. I also forgot to mention that this is a picture of what mannitol salt agar looks like. It's less red than blood agar. You can always tell blood agar when you see it. It has a very distinctive sort of cherry red look. This agar is much more, it's almost a pinkish color. It has a much lighter color to it when you see it. Now let's talk about differential media. Remember, selective media can select which organisms are going to grow one species over another species. What differential media is going to do is differentiate between strains of bacteria. And it does that by telling us with color changes whether or not a particular metabolic reaction is going on in that bacterium. Again, it tends to use indicator dyes, which will change color when pH conditions change. So remember what a strain is. We talk about a genus and a species, but then within a species, there can be different strains. We said it's just like the way we talk about breeds of dogs. All dogs are Canis for their genus and Familiaris for their species. But we all know that there are different breeds of dogs, and they look very different and they behave very differently. That's analogous to a different strain of bacteria. Think of it like a breed of bacteria within a species. So one of the most common differential media is one called MacConkey agar. And what MacConkey agar is going to allow us to do is both select and differentiate certain kinds of bacteria. So MacConkey is both selective and differential. On the selection side, it's going to select for bacteria that are gram-negative and live in the intestinal tract. So enteric bacteria. Anything that's not gram-negative and enteric is not going to be able to grow on MacConkey. That's how it is a selective medium. It's also differential. And what it's going to differentiate between is gram-negative enteric bacteria that can ferment lactose from those that can't. So if it can grow on MacConkey agar, if we get something to grow on MacConkey agar, we know it's a gram-negative enteric bacterium, but there are lots of those. So how do we differentiate what kind we have? Well, some of them will be able to ferment lactose and some of them won't. Now, how are we going to know one from the other? We're going to look for a color. Bacteria that can ferment lactose are going to produce acid products, acid metabolic products or waste products. And what will happen is when they grow, they're going to be pink on this agar. The colonies themselves are going to grow as pink colonies. Any bacteria that are non-fermenters, that cannot ferment lactose, well, they're not going to secrete those acid products. So they're going to grow as pale or colorless colonies. So we can look at a MacConkey plate, and if we've got things growing on it, we can look at the color of those colonies and we can know a lot about this organism. If it's growing on MacConkeys, we know that it's gram-negative and it's an enteric organism. It lives in the intestine. If it grows pink, we know it can ferment lactose. If it grows white or pale, we know it cannot ferment lactose. And that's going to help us narrow down what kind of an organism we're dealing with. So McConkey is one of the agars that is both selective and differential. Blood agar is another good one. Blood agar, as I said a few minutes ago, is complex. Remember, we can also refer to those as enriched media. One of the ways that blood agar is used is to differentiate strains of Streptococcus, one from the other. So Streptococcus, like lots of bacteria, will grow well on blood agar, but only some strains of Streptococcus are able to do what's called hemolysis. They're able to break down red blood cells. They make a molecule called hemolysin. If you translate this word, if you break this word down, it means blood breaking. So if a bacterium produces hemolysin, it's able to hemolyze or break apart red blood cells. And what happens is when they grow on blood agar, the colony is going to be surrounded by a clear zone where the blood cells are gone because they've been broken apart. So blood agar, you could think of as complex or enriched. Both of those terms mean the same thing. And it's differential. It can differentiate strains of Streptococcus that produce hemolysin. So when you work in a microbiology lab and you work with all these different kinds of media, you start to learn what each type of media either selects for or differentiates between. As we move forward in the semester, we're going to practice, we're going to do experiments using different types of selected media so we can learn what they do and what they're used for. And we'll look at two kinds of selective media today. All right, so for our 6 types of media, we said basal and enriched, selective and differential. And the last 2 is transport and storage. Now, we already talked about transport media. We said transport media is going to be a medium used to literally transport samples from the place where they're collected to the laboratory. So it has enough nutrients in it to support these cells and keep them alive until they get to the lab. Transport media is not fancy, and it's not designed to encourage a lot of growth. It's just going to keep the cells alive if there are cells in the sample. Storage media is very different. What storage media is used for is freezing bacterial samples or other microbial samples. So it has a special kind of molecule in it called glycerol. And glycerol is protective of cells when they're frozen. If you know anything about the problems with freezing living things, it's not the freezing that kills a cell, it's the thawing that kills a cell. It's just like, it's not the freezing that will ruin your pipes. It's the thawing that will break open your pipes under your house. When water freezes, a little more chemistry, when water freezes, it expands. So what happens in cells when you try to freeze them is the water, as it freezes, it expands and it breaks the membrane, it breaks the cell. And then when the cell thaws, now you have, just bits and pieces of a cell left. When you add glycerol to the cells, they will replace the water inside of them with the glycerol. And glycerol doesn't have the same behaviors that water has. So we literally get these microbial cells to take in glycerol, and it will fill up the cell and replace the water in the cell. And then when you freeze them, you can thaw them and keep the cells alive. So that's called storage media. All right. Any questions about those 6 types? It can be a little hard to remember the difference between a selective media and a differential media. What I tell students to try to remember is selective media is going to select or pick which things can grow and which things can't. Differential media is going to differentiate or tell the difference between 2 very closely related organisms, 2 different strains of bacteria. Any questions so far? All right, I'm going to jump back in then. So we're going to do an exercise together now. And, you know, through the magic of computers and the internet, we're going to be able to do all of this together in like half an hour because all of this has actually already been done. So we're going to use a series of bacteria for our experiment today. You'll notice I'm going to call them bugs. I like to call bacteria bugs. These are the bugs we're going to use. We're going to use Escherichia coli. Remember, that's what E. coli is scientifically. We're going to use an organism called Enterococcus Fecalis. We're going to use Staphylococcus epidermidis and Staphylococcus aureus. Now, we already know a little bit about these bacteria. These are well-studied bacteria. E. coli is a gram-negative rod. That's its gram status. And it is an enteric organism. And like I said a minute ago, we use that term to describe any organism that lives in the enteric tract or the intestinal tract. So another term you'll hear is fecal. People will just call these fecal organisms because you can often find them in feces. Enterococcus fecalis, you can tell a little bit about this organism from its name, right? Entero, meaning intestinal, coccus, that's its shape, and fecalis is a fancy word to say fecal. This one is gram-positive, so it's a gram-positive coccus that lives in the intestine. Enterococcus is considered a fecal streptococcus. Now that's unfortunate language because it's not streptococcus, it's Enterococcus. But when people call this a fecal streptococcal organism, what they're saying is it looks a lot like a streptococcus, gram-positive coccus. Staphylococcus epidermidis is a gram-positive coccus that lives on the skin. And Staphylococcus aureus is also a gram-positive coccus, and it lives on the skin as well as in the nose and in the back of our throats. Now, I want to say something really important here because this is something, a really important thing to know about organisms like these. All of these organisms have the ability to be pathogens. In other words, there are strains of E. coli that are pathogenic. There are strains of Staph aureus that are pathogenic. But there are other strains of all of these organisms that are not pathogenic. In fact, something like 15 or 20% of human beings walk around with Staph aureus all over them, on their skin, up their nose, in their throat, and the staph doesn't bother them at all. The staph doesn't cause any problems in them at all. And then there are other humans walking around who, if they got Staph aureus in the back of their throat, they would get sick. So you have to always remember that even when you see a name that looks like a pathogen, like a lot of people hear E. coli and they think about, a pathogen. There are strains of E. coli that are pathogenic. There are very dangerous E. coli pathogens, but there are other strains that live inside of all of us and are an important part of our microbiome. So you can't tell just by looking at the name of an organism whether or not you've got a pathogen. You have to know a little bit more about it. So there are 4 organisms, we'll say, at play today. Here are the 4 organisms. And then what I did for our experiment was I chose 2 of them secretly, and I mixed them together in a container. Here's our goal. We're going to do a little bit of culturing with special media, and we're going to try to tell which 2 of these organisms I chose. All right, we're going to see if you can figure that out. I told you the last time we were together that gram staining is super common, and we use gram staining sometimes to help us tell the difference between big groups of organisms. So, if you look at this information that we have about these bugs, you can use gram staining, right? We can use gram staining to tell whether we have E. coli, because E. coli is the only one that should be a gram-negative rod. So, if I did a gram stain of my mix of 2 bugs and one of them was a gram-negative rod, I'd know that it was E. coli. It's the only gram-negative rod out of the bunch. But I can't use gram staining to tell the other ones apart, can I? Because they're all gram-positive coccus, gram-positive coccus. So I need something else. I need another bit of information, another way to tell them apart. So I've got a container that has 2 of those organisms in it. We don't know which 2. Now let's talk about the media we're going to use. We're going to use a basal media, an agar called nutrient agar. And we're going to use Mannitol Salt agar. Now, I told you a few minutes ago that Mannitol Salt agar is selective at 7.5%, which is what we'll use. It selects for Staph aureus and Staph epidermidis. Both of those things can grow on it. It selects against virtually all other bacteria. In other words, virtually all other bacteria cannot grow on this. Remember, there is a little exception. We said Enterococcus sometimes can grow weakly on it, but these are the 2 that grow primarily on this agar. Now, I told you that MSA is also differential, and here's how it's differential. Mannitol Salt agar can tell the difference between strains that can ferment mannitol and strains that can't. So just like I said a few minutes ago with the McConkey agar, I said McConkey can tell lactose fermenters from non-fermenters. Well, MSA can tell mannitol fermenters from non-fermenters. Both of those are just carbohydrates. They're just sugars. Now, S. aureus, Staphylococcus aureus, and Staph epidermidis are different in their ability to ferment mannitol. Mannitol fermenters are going to produce an acid byproduct, and the media is going to turn yellow. Now, this time it's not the colonies that are going to be yellow. The agar itself is going to turn yellow. if the organism growing on it can ferment mannitol. If the organism can't ferment mannitol, the media is just going to stay pink, this pinkish red color. So Staph aureus can ferment mannitol, but Staph epidermidis can't. So by looking at the color of the agar, I'm going to be able to tell which one I have if I get one of them to grow. This is what that looks like, by the way. Here's again mannitol salt agar that has not been inoculated. There's no cells on this, and it has that very light pink color to it. Over here on the right, there's some Staph epidermidis streaked across the top of the plate and some Staph aureus streaked across the bottom of the plate. And this plate has been in the incubator for about 24 hours. So if you look closely, you can see there's cells growing on here. I just made streaks, so there's no individual colonies here. There's just these lines full of growth. But I think you'll agree that the agar has turned yellow down here because Staph aureus, when it grows on this media, it ferments the mannitol and it creates an acid byproduct that changes the pH and changes the agar to yellow. Staph epidermidis can't ferment the mannitol. It doesn't know what to do with it, so it just leaves it alone. It's growing, but it's not fermenting the mannitol. So the agar stays that nice pinkish color. Both organisms can grow on mannitol salt, but I'll be able to differentiate them by the color of the agar. So we'll use mannitol salt. We'll take our unknown combination of organisms and we'll spread it on some mannitol salt agar. And the other thing we'll use is called eosin methylene blue agar, or EMB. I'll show you a picture of EMB in just a minute. But EMB is selective for E. coli and other what are called fecal coliforms. Have you ever heard this word coliform before? You ever heard that term? That's a very commonly used term in medicine. People talk about fecal coliforms. What they're talking about is E. coli and organisms that are closely related to E. coli. They live in the intestine. Some of them, some of the strains, are pathogens. Others are not. Others are important parts of our microbiome. So if you ever hear the term coliform, fecal coliform, they're generally talking about E. coli. So it selects for E. coli. It selects against anything that's gram-positive. So remember, when you look at our 4 organisms, we have what? 1, 2, 3 gram-positive organisms as our possibilities, right? They're not going to be able to grow on EMB. The only thing that can grow on EMB in this list is E. coli. So we'll be able to use EMB to select for E. coli if E. coli is one of our 2 unknown organisms. Now, EMB is also differential. It's going to differentiate 2 kinds of fermenters here. There's 2 kinds of fermentation action going on here. If the organism can ferment lactose and or sucrose, either one of those, they will grow as purple colonies. And they will have this really amazing green metallic sheen to them. I'm going to show you a picture of this. It's very distinct looking. Sometimes those metallic looking purple colonies actually have a dark dot in the middle. They call this a fisheye look. Again, I'll show you a picture in a minute. There are other fecal coliforms that can grow on EMB, but we know we have E. coli. We know we have E. coli if we get that greenish sheen. Other fecal coliforms include organisms like Salmonella and Shigella. You may have heard of those. Neither Salmonella nor Shigella can ferment lactose or sucrose, so they're going to grow as colorless colonies. So if we get growth on EMB, we have to look at it and see what color the colony is and whether or not it's got that greenish look. Take a look here. This is what EMB looks like when nothing is on it yet. It has a very distinctive color. Now, some people call this purple. I guess it's like an eggplant purple, maybe. I don't know if I see purple or if I see more of a maroon color here, but it has a very distinctive dark color, okay? Now, when you put E. coli on EMB, this happens. This is one of those quadrant streak plates we talked about, where you put a lot of cells on first, and then you pull some of those cells into the next quadrant and you streak them. And then you pull some into the next quadrant and you streak them, and you try to get individual colonies to grow. They were successful on this plate. There are definitely individual colonies growing here. They're very little, but there are colonies growing here. But in this case, I really want you to look at over here, especially. Not so much at the colonies, but over here. Look at that green color. It is metallic when you look at it under the light. It absolutely shines like it's metal, like it's got some kind of metal in it. It's a very distinctive green color. And the only organism that's going to do that is E. coli. There are other organisms that will grow on EMB, but they're not going to give that green color. This is what they're going to look like. So this is just a big old smear of bacteria in this quadrant. That's all that is. That line is there because somebody took just a loop full of cells and just smeared it right there. And it grew as this big, thick line of cells. You can see another line here that grew and another line here that grew. Over here, there's nothing growing. So remember what can grow on EMB. E. coli and other fecal coliforms can grow. And then there are some other enteric organisms that can grow, like Salmonella and Shigella. We can differentiate all those different fecal bacteria by looking at how they grow. This is E. coli because only E. coli gives that green color. This is a fecal coliform because it's growing as a purple colony without any sheen on it. So it's a gram-negative fecal organism. It's another coliform, but it's not E. coli. And finally, we have this line right here, which I know is a little hard to see. This one doesn't have any color. It's just sort of clear looking. This could be Salmonella or Shigella. This organism can ferment either sucrose or lactose. This one can't. And this one's E. coli. So lots of information for EMB, I know. Lots of information about what is what on EMB. But it's primarily used to find E. coli. This is what I meant when I said sometimes you get this effect. Look at these colonies. Here's an EMB plate that was quadrant streaked. They started here, then they pulled some over here, then they pulled some over here, and then they pulled some over here. And in the last quadrant, they got individual colonies to grow. It's hard to see over here, but these colonies are purple and they have a metallic sheen. Remember, always look back at the first quadrant to see the sheen. But look at the colony. Look in the center of these colonies. There's a black dot in the center. For some reason, some strains of E. coli do this. They produce a little black dot in the middle of their colony. It's part of their colony morphology. People call these fisheye colonies. Not all E. coli will grow like this, but some of them do. So on EMB, we can grow gram-negative organisms. We can't grow, no gram-positives will grow, but gram-negatives will grow. Mostly fecal organisms, gram-negative fecal intestinal organisms. If it's E. coli, it's going to be a purple colony that has a metallic sheen to it, a green sheen to it. If it's another kind of fecal coliform, not E. coli, if it's another kind, it'll grow as a purple colony. It just won't shine green. If it's another fecal organism altogether, like Salmonella or Shigella, it'll grow clear on EMB. Now, here's a question I want you to think about for a minute. I told you a couple minutes ago that we're going to grow our mystery culture that has 2 of our 4 organisms in it. I said we're going to grow on nutrient agar, we're going to grow on Mannitol Salt agar, and we're going to grow on EMB. And we're going to see if we can use all of the results to figure out which 2 organisms are in our container. Why am I growing it on nutrient agar? Why would I bother with that? Nutrient agar is not selective or differential in any way. Nutrient agar is a basal media. It's very basic. Lots and lots of things can grow on nutrient agar. Why in the world might I do that?
Are we trying not to rule out at this point the gram-positive and gram-negative?
Well, both would grow on nutrient agar.
That's kind of what
it's not going to give me any information, is it? It's not going to help me tell. Any ideas why you might do that in an experiment like this?
Is it a control?
It's a control. What's it going to tell me?
It's going to tell you if there's anything growing.
That's exactly right. Good job. It's a control. I'm going to put all 4 of those possibilities onto nutrient agar. And the reason I'm going to do that is because I need to know that all 4 of them are alive and well. All 4 of them are able to grow. The last thing I would want to do is, choose one of those 2 to put into my unknown and have it not be able to grow at all. So sometimes when you're in the laboratory and you're working with living organisms, the very first thing you need to do experimentally is make sure they're still alive, right? Make sure they can grow. All 4 of those, all 4 of our possibilities. I'm going to try to grow on nutrient agar. And that way I know for sure that they're able to grow. Those cultures are alive. So I can pick 2 of them and I can use them in my experiment. Good job. All right. So I drew 4 quadrants on the bottom of the nutrient agar plate. That's the Na plate. And using my very best aseptic technique, I just made a streak, a simple streak of each of those 4 pure cultures. 1 culture into each quadrant. I don't need to grow isolated colonies here. I'm not trying to get colony morphology. I don't need to make spread plates. I'm not trying to count colonies. I just want to make sure that all 4 of my cultures are alive. I'm going to streak that nutrient agar plate. I'm going to put it in the incubator, upside down, of course. at a very commonly used temperature, 35 degrees centigrade. Most bacteria can grow at that temperature. And I'm going to leave it there for 24 hours, which again is just the very basic starting incubation time. And then I'm going to examine the results. Remember, this is just to make sure my cultures are alive. Now, these are two different plates, but the idea is the same. You can just see that there's lines drawn on the plate. And then a very simple spread, just a very simple streak of each organism, one in each quadrant. So I'm not going to get colonies here. You're not going to get colonies unless you quadrant streak or you make a perfect spread, like with a hockey stick. You're just going to get this sort of a mass of things growing, a mass of cells growing. Here's another way to do it. You can see on this plate, these are student plates, by the way. Some students made these in class. This one, the student just did the streaking a little bit differently, just did a sort of an S streak. That's fine. We're just trying to see if the cells can grow. And the answer is yes. All 4 of our cultures grew on the nutrient agar. So since I know all 4 of our organisms are alive and well, I'm going to pick 2 of them and put them together in a container. And now we're ready to finish the experiment. Part 2 here, using the unknown, we're going to make 1 quadrant streak on MSA and 1 quadrant streak on EMB. Both of these augers, remember, are selective and differential. And we're going to put both plates upside down into the incubator, 24 hours, 35 degrees centigrade. And we're going to examine the results. Now, the reason I did a quadrant streak is because quadrant streaks are hopefully going to give me individual colonies. And remember, for these, the colony color may matter, right? So I need some colonies to grow as well as just some mass of cells to grow. All right. 24 hours later, here is our mannitol salt. Now, there's a line here. I want you to ignore this line. I took the combined culture and I did a quadrant streak. The reason the line is here is because, again, a student made this plate. And it's just easier when you're learning to do a quadrant streak to draw the quadrants. It's just easier. But remember what this student did. The student took a loop full of the unknown combined culture and put it in the first quadrant, just smeared it into the first quadrant. And then cleaned the loop and then drew the loop through that into the second quadrant and spread it out. In other words, just pulled a few of these cells into the second quadrant and streaked them out. And then cleaned off the loop again in the incinerator, then drew through that quadrant, started to spread them out, and then drew through that one and spread them out. Now you'll notice we didn't get growth in all quadrants here. That's okay. That's not a problem. We're trying to identify an organism here. So the important thing is, did you get any growth? And the answer is yes. Look, there's colonies growing on here. Some of them are all jammed together, but that's okay. There are colonies growing on here. So we already have a result here. We got something to grow on mannitol salt agar, and it turned the agar yellow. Here is our EMB plate. Bummer. Absolutely nothing grew on this EMB plate. That's important information though, right? EMB is selective. Some things can grow on it, some things can't. Now, how do we interpret this? Remember, we have 4 possible organisms. 2 of them have been combined and put on these plates. So what we need to do is figure out one by one, is it possible that these organisms are in the unknown combined culture? Let's think about E. coli first. Could E. coli be one of those 2 unknowns? We know that on Mannitol Salt agar, there are only a couple of organisms that can grow. It's got a lot of salt in it. E. coli is not one of those. So I'm going to put a check mark here. E. coli cannot grow on MSA. Now you might say, well, why are you putting a check here? Well, maybe I now know that this is not E. coli. Whatever's growing here is not E. coli. Now, E. coli could be the second organism in the culture, right? There's 2 organisms in that unknown. So maybe E. coli is the second organism. It didn't grow because it can't grow on MSA. But I know for sure it's not the one that's growing on MSA because it can't grow on MSA. Does that make sense to you? Only 2 things can grow on MSA, really, with an exception that I mentioned. But Staph aureus can grow. Staph epidermidis can grow. Both of those are possibilities for our unknowns, right? E. coli can't grow on MSA. I got 1 organism to grow on my MSA plate. I know it's not E. coli because E. coli can't grow on MSA. But I could still have E. coli in my unknown because maybe it's in there and it just didn't grow, right? I can only see 1 organism growing on the MSA plate. 1 organism grew. This one right here. There's no second kind of colony anywhere, just these 1 type of colony growing. The one that is growing as these little yellow circles. This is 1 type of colony, 1 organism growing here. It's possible E. coli is still a possibility, in other words, because I spread 2 organisms on this plate. This one grew, the other one didn't. So maybe the other one is E. coli. You see what I'm saying? I don't know yet. I don't know yet. E. coli is still in the running. But remember this, nothing grew on the EMB plate. E. coli grows really well on EMB. E. coli is going to grow purple and it's going to have that beautiful metallic sheen on it. If E. coli is one of my 2 organisms, it should have grown on the EMB. And it didn't. Nothing grew on the EMB. Neither one of the 2 unknown organisms grew on the EMB. So I don't have E. coli. It makes sense that E. coli would not have grown here, but it doesn't make sense that it didn't grow here. So I can rule out E. coli now. Does that make sense? Does that make sense to you? How I can now say, oh, I don't have E. coli.
It does to me, yeah.
I would expect E. coli to grow on EMB, and it didn't. Nothing grew.
Yes, it makes sense.
Okay, good. All right, let's look at our other organisms. Could it be the E. fecalis organism? I don't expect E. fecalis to grow on MSA. So once again, maybe E. fecalis is the one that's not growing on MSA. Here's another important piece. I do not expect E. fecalis to grow on EMB. E. fecalis is a gram-positive organism. We said EMB selects against gram-positives. I got 2 check marks. I have 2 checks. I didn't get growth here, but I didn't expect growth for E. fecalis. I didn't get growth here. I didn't expect growth for E. fecalis. Looks like E. fecalis may be one of our organisms. But let's keep going. Could Staphylococcus epidermidis be one of our 2 bugs? Well, we know it grows well on MSA. It could be this one, right? Something growing on MSA. But we also know that S. epidermidis cannot ferment mannitol. So the agar should stay pink. This is obviously turning the agar yellow. So this cannot be S. epidermidis. I didn't get S. epidermidis to grow on the EMB, but that makes sense because it shouldn't grow on the EMB. So I know it's not S. epidermidis because of this, because this organism is fermenting mannitol and turning that agar yellow. Could S. aureus be one of our bugs? Well, S. aureus also grows well on MSA, and S. aureus does ferment mannitol and turn the agar yellow. There you go. This one here, this is S. aureus. It's really the only organism that will both grow on mannitol salt and ferment the mannitol. S. aureus cannot grow on EMB because it's gram-positive. Good, it didn't, right? It didn't. So I'm going to conclude that my combination was E. fecalis and S. aureus. The E. fecalis didn't grow on either plate because it shouldn't have grown on either plate. The S. aureus grew on this one and turned it yellow, as it should have, and it didn't grow on this one because it shouldn't have grown on this one.
So could I just ask one question?
Yes, please.
The E. fecalis, we know that it's there because it grew in one of the quadrants, but how do we know it was E. fecalis that grew in the quadrants of the control?
Oh, well, so we had 4 pure cultures. And
Oh, okay, so you just did 4 separate ones.
remember what that means to us. When you hear me use that word, I have a pure culture of something. What that means is I took some of those organisms like out of the freezer and I grew them purposefully in culture. So I had 4 cultures growing. I spread each one in a quadrant on the nutrient agar, so I knew all 4 cultures were alive. And then I secretly picked 2 of them and I smeared them onto the mannitol salt and onto the EMB. The reason we know one of them is E. fecalis is because we ruled out the other 2, right?
Right.
We ruled them out. Remember, we're saying it's E. fecalis because we didn't grow anything. We didn't grow any E. fecalis because we shouldn't have grown any. And the other ones should have grown on one or the other plate. So it's really, you're really making that decision based on ruling out everything else. The Staph aureus is different. The Staph aureus we did grow, and it grew correctly. It grew the way we expected it to grow. But yeah, a lot of times what we're left with is a negative result. And as long as we're confident that, you know, we've ruled out the other possibilities, we can be sure about that negative result being one specific organism.
But it's just process of elimination.
Yeah, it's process of elimination. That's exactly right. Now, if I suspected that I had, let's say this wasn't an experiment, let's say I had a sample come in from a patient or from the environment, and I wanted to find out if E. fecalis was growing in it. I wouldn't stop here. I would find a media that E. fecalis could grow on. I would look for a media that could select or differentiate E. fecalis. And I would try to grow it on that just to confirm for myself that it was E. fecalis. But among these four, those four specific organisms, I've ruled out the other ones. So it has to be. It's a puzzle. It's a little bit of a puzzle to solve. Now on this slide here, towards the end of the slide set, I've just reiterated some of the things we know about these bacteria while you're, if you go through and review this video later. Remember, 3 of these organisms were gram-positive, so they were going to be hard to differentiate from each other. The E. coli is a gram-negative, so frankly, we could have used gram staining to differentiate too, but we use the 2 selective media instead. We know that E. coli and Enterococcus both are these fecal kinds of organisms. And we know that Staph aureus and Staph epidermidis are these sort of skin organisms or skin and nose organisms. So they're kind of related to each other, but we were able to differentiate them based upon some of their metabolic abilities. Who could ferment what? And how did that make them grow or not grow? Remember, for EMB, it selects for E. coli and other, quote, fecal coliforms, this sort of generic word we use for anything that's closely related to E. coli. But even more importantly, it selects against anything that's gram-positive. And Enterococcus fecalis is gram-positive. So these anything gram-positive cannot grow on EMB. So if we get something growing on EMB, we already know a lot about it. We know it has to be gram-negative, and it has to be one of these fecal coliforms. If it's E. coli, it's going to have that green glow. The mannitol salt, remember, the mannitol salt is all about that salt. There's only a couple things that can grow in salt: Staph aureus, Staph epidermidis, and weakly, very weakly, some Enterococcus, but I told you not to worry about that today. So if I see growth on mannitol salt, I'm automatically thinking, oh, it's Staph. It's either Staph aureus or it's Staph epidermidis. And the way I tell those two apart is by the yellow. If it's growing yellow on mannitol salt, if it's turned the agar yellow, now I know I have Staph aureus, because Staph aureus can ferment mannitol and Staph epidermidis can't. It doesn't know what to do with it. So it just doesn't use it. And the agar stays pink. All right. So our first little exercise in, differentiating one organism from another based on how they grow and what they do when they grow. That's a lot of what's happening in a microbiology lab. When we get these samples in and we find we have organisms, but we don't know what they are. The first thing we're going to do is gram stain. We're going to try to grow them in culture, and then we're going to start putting them onto different kinds of media and seeing how they grow and seeing whether they grow and what color they grow. And we start getting a list together of all the features of this organism, and it's going to help us figure out exactly what it is that's growing. Exactly.
Is that how they actually do it in hospital labs? I mean, if you go in for a strep throat test, is that why it takes 24 hours?
Yeah, because we have to grow the culture first, And then we plate the the strep. And so we'll gram stain, and then we plate it, and we can do some tests for strep. Yeah, so the doctor gets that immediate idea, remember, with the rapid test, and that's about antigens. We're going to talk about that later in the semester. But the reason it takes that 24 hours to get your culture results back is because we have to grow it up in culture first. So we can plate it on these differential and selective media. That's exactly right. So the doctor puts you on antibiotics based on the rapid test, but we always want to confirm it. We always want to confirm it in the micro lab. All right, good questions today. Any other questions? All right, when we get together on Wednesday, our next exercise, we're going to look at a different kind of media. It's very specialized. And it's not a selective media or a differential media so much as it's a type of media that's going to tell us a lot about what kind of oxygen requirements unknown organisms have. It's going to tell us whether or not they love oxygen and they use oxygen for their metabolic processes, or if oxygen is toxic to them, or if they can use oxygen, but they don't have to use oxygen, and so on and so on. So it'll be all about oxygen when we get together on Wednesday. Don't forget, you have this biochemistry lecture and the growth lecture. The biochemistry lecture might take a little extra time, so be sure to give yourself that this week, and keep a list if you have any questions. Okay? Anything else? All right, ladies, that's all I have for you today. I will see you next time, okay?
Thank you.
You have a good night.
Thank you.
You're welcome.